I want to shift from the molecular to the molar and talk about stress and hormesis in the whole behaving animal. I'm going to present a recent line of research from my laboratory on stress resilience, and in the process of doing so, identify some preconditioning and postconditioning manipulations that dramatically increase the range of adaptive responding under very stressful circumstances. Hormesis has not had a big impact on stress research in psychology. Many of you have actually talked about the barriers, siloing and the barriers to clear communication among disciplines. That problem is also inherent within psychology. Psychology is such a broad area that we create our own little silos, and half the time when my colleagues give a presentation on stress, I have no idea what they're talking about. And I think part of the problem here with the lack of an impact on hormesis on psychological thinking, and I think it's going to have an extremely important impact in the very near future, is that the psychology of stress research actually evolved independent of neuroscience. And it's only now that those areas are actually starting to merge. I think Ed has done a lot to actually break down the barriers among areas, and both by sponsoring conferences like this, which I've had a great time at, as well as publishing a number of papers that emphasize the importance of this idea of hormesis. For instance, in 2007, the first paper that I read of his, he published a paper with 57 of his close colleagues. And in that paper, they identified a number of biological phenomena that, in which pre-exposure to small stresses build resilience to large stresses. And many of those phenomena actually show this bitonic or biphasic dose-response curve. And in that paper, he identified the Yerkes-Dodson law, which is a mainstay of psychology, as one of those phenomena. And then a year later, he published a monograph where he expanded on that idea. So I think that's a good jumping off point. How does this thing work? That's that. So I think good jumping off point to start to understand our work. So the Yerkes-Dodson law has been with us for a long period of time, since 1908. It's a biphasic function that describes task performance as a function of stress, anxiety, or arousal level. Performance generally improves over a wide variety of tasks as stress levels increase, and then declines as stress levels exceed an optimum. The optimum level of stress increases as task difficulty increases. The original study was done in rats, but it's been replicated in just about every species since then, including humans. Picture's worth 1,000 words. So this is basically a dose-response curve plotting task performance against stressor severity for a simple task in red and a complex task in blue. So the simple task here, performance generally increases over a wide range of stresses until it reaches an optimum. At more severe stresses, actually performance falls off. For more difficult tasks, the optimal stress level is considerably lower. Performance increases as stressor severity increases to that optimum and then falls off. My lab deals with stress levels way to the right of any of these functions, where nobody or almost nobody performs optimally. The stressors here are severe, uncontrollable, and traumatic. And for most of my career, I've actually tried to understand the pathology that actually emerges from experience with traumatic stress. In psychology, for humans, the most common disorder is post-traumatic stress disorder. We can model that in rats by exposing them to traumatic stress, and they will develop some of the symptoms that you see in a human. So given this Yerkes-Dodson context for our research, when we started this research, the general idea was to find preconditioning or postconditioning manipulations where the conditioning event actually is this trauma, where we could increase the range of adaptive responding so that even here at extraordinary levels of stress, the organism would perform adaptively. The lab starts with rats in a rodent model of post-traumatic stress disorder, and we then try to do translational work where we work with, this is a United States Marine. We've done 2 studies with the Marines, one with Marines pre-deploying to Afghanistan. We're trying to increase their resilience and decrease the incidence of post-traumatic stress disorder. More recently, we've done research with Marines going through the basic recon course at Camp Pendleton, which is just down the road from UCLA. If you're a psychologist and you want to do stress research, it's actually very difficult to do in the laboratory, mainly because humans don't like to be stressed, and IRBs don't let you stress them. The nice thing about working with the military is they stress themselves, and they expose themselves to severe stress. All we have to do is show up and actually record their behavior. We've also more recently started working with trauma patients. So this here is a trauma patient that's about to enter the LA County USC Trauma Center, which is the largest trauma center in the country. And we now have a study ongoing where we're trying to prevent the development of post-traumatic stress disorder in trauma patients. Excuse me. So I'm going to turn back to rat research. The procedure we use actually to induce PTSD-like symptoms in a rat is very simple. It consists of 2 phases. Phase 1 is exposure to a traumatic stress in the form of 100 1 milliamp unsignaled inescapable tail shocks in a restraining tube, or simple restraint. That's simply an apparatus exposure control over about 2 hours. Okay, the shocks are painful. It's not the painful stimulus that induces the adverse response. Those painful shocks induce a very strong fear response. We keep the animal in that fear response for over 2 hours, and no one is made to be afraid for that period of time. 24 hours later, we test the animal in a shuttle box, and I'll describe that in just a minute. We're looking for 2 responses. One, we measure behavioral freezing, and behavioral freezing is a measure of fear. PTSD patients show significantly heightened arousal, sympathetic activation, and fearfulness. At other times, they actually fail to respond to stimuli. And so we measure escape latencies here, and I'll describe that in a minute, which we take as a measure of conservation withdrawal. Conservation withdrawal is a term, actually an old term from psychiatry. There was a couple of psychiatrists in upstate New York who had a patient who was institutionalized with severe anxiety disorders. She had real problems with her mother. And when they come in for therapy and they say, okay, Mary, let's talk about your mother, the initial response to that was extreme anxiety and agitation. She would then transition into a state where she would not respond to stimuli. Her cognition was garbled, and she'd show behavioral depression. And what Engel and Schmale argued is that the sensory unresponsiveness, cognitive dullness, and behavioral depression that this woman showed was an adaptive response to an extreme metabolic challenge. And what those responses did by shutting down, they husbanded limited energy resources and facilitated the recovery of energy and homeostasis. And what we'll see for our animals that have been traumatized, they will show extreme deficits in escape performance. Basically, they're getting foot shocks, they're not moving. And we have since linked that failure to escape to an increase in adenosine signaling in the ventral medial striatum. And so we went back and borrowed this idea of conservation withdrawal because it actually nicely characterizes the behavioral state that these animals are in, and it links the failure to respond to brain metabolics. This is the restraining tube where the animal gets shocked or simply restrained. 24 hours later, it's tested in a shuttle box, and we measure 2 things here. We start with 5 easy trials. If the rat is on this side of the box, it's got to run to this side of the box to turn a foot shock off. We then wait 60 seconds, and during that 60 seconds, we score behavioral freezing as a measure of fear. We then shock it 5 times, so we get a measure of fearful over 5 trials. Then we shift to a more complicated task contingency. If the animal is on this side of the box. It's got to run on this side of the box and then back again. And when we expose the animal to the more difficult task, it transitions from this anxious, agitated state that it enters the situation in to this state of conservation withdrawal. And these animals show severe deficits in escape performance. So here's our two basic measures for just a shocked and a restrained group. The shocked group for all these slides will be shown in red. The restrained groups will be shown in blue. Okay, and here, this is behavioral freezing. These animals are showing considerable more fearfulness right away that persists over the entirety of that first part of the task. These are the shuttle escape latencies. The worst an animal can do on an escape trial is actually 40 seconds. If it doesn't make the appropriate response in 40 seconds, we turn the shock off and go on to the next trial. So here the animals in red, the ones that got the traumatic shock the day before, are averaging around 30 seconds, which means that of this group of 8 subjects, almost all of them are failing to escape and taking 40 seconds of shock on the trial. Okay. And so here, I'm going to define resilience as the ability of some kind of manipulation actually to mitigate the effects of traumatic inescapable shock on later fear conditioning and escape deficits. And so this is Tracy Plum and Patrick Cullen who did the work on the paper, and they're both now doctors and off on postdocs. And I don't have to explain hormesis to this group. Okay, so here's the basic paradigm here. We take the idea that exposure to comparatively mild stresses should build resilience to more traumatic stresses. So we started off here with 6 groups. These are our basic traumatic stress groups. These are restraint control and our traumatic shock group. And then other groups got some preconditioning stress. The little r here indicates 30 minutes of restraint stress. So we just stuff them in the tube for 30 minutes. And then there's a lot of theory in the psychological literature that for something to be beneficial, to actually build resilience, the animal has to recover its baseline, its stress baseline, before the next stress comes along. And so in this experiment, we either gave them 3 days of restraint stress or 5 days of restraint stress, and all of those had an interpolated day of rest. Okay? And so that's our pre-treatment shocks. Here are the test data and freezing. So here are the animals that just got the traumatic stress, no preconditioning. The blue squares are the restrained group that got no preconditioning. And everybody else is somebody that got some preconditioning. And here for the freezing data, everybody did just about the same. So, and substantially better than the untreated control. The escape latency is a little bit clearer. Again, the group that got just the traumatic stress is performing abysmally on the task. The ones that just got the restraint are the blue squares. They're right in there. And what these data suggest, and the important comparison here, is the red triangles. Is this done? Or I'm just missing? Oh, the red triangles versus the red circles. So that's 3 days of restraint stress. This is 5 days of restraint stress. And it's pretty clear that 5 days of restraint stress is better than 3 days. Okay, so more is better. In the next experiment, we looked at the pattern of restraint stress, and the question here is interpolated rest actually a necessary condition to see a benefit of this procedure? So we have our restrained and shock groups as our basic trauma comparison. This group got 3 days of restraint stress without interpolated rest. This this group got 3 days of restraint without interpolated rest, and then 3 days off. Got vacation, big weekend. And then the third group here is the group that got 3 days of restraint stress with interpolated rest. And these are the shock data. And basically what we're seeing is, for the freezing data, that it doesn't matter. The pattern of actually restraint stress doesn't matter. Everybody who got the preconditioning stress actually performs quite well. And this, here for the shuttle escape latencies, we basically see the same thing. Okay. All right. Now the question is what's a mild stress? And so here what we're going to do is actually increase the severity of the stress here. A little s here is being placed in a restraining tube and getting 30 inescapable tail shocks. Okay. We know that one session of 30 inescapable tail shocks will actually produce our exaggerated fear responding. It will not produce our conservation withdrawal responding. Okay. So here we're looking at both the pattern and the severity of the stressors. So here these guys got 3 stresses in a row prior to getting shocked. Everybody's getting shocked here except for the restrained group. 3 days of shock and then 3 days off, and then the procedure where they get shocked, interpolated rest. Why can't I do this? Shock, interpolated rest, and so forth. And under those conditions, you get a mess. So we'll start by looking at the red squares and the blue squares. That's our basic trauma difference. If you got 3 days of mild shocks, moderate shocks here before the big traumatic shock, it actually looks like it makes you worse. I mean, we're sort of at a ceiling here. If you got 3 days and then 3 days off, you find some improvement, and the most effective condition here is actually getting 3 days of small shocks and interpolated rest. These are the escape latencies. Actually, these are a little bit clearer. Again, if you got 3 small shocks in a row before the traumatic shock, you're performing at least as badly as the unpreconditioned shocks, so you're getting no help from this. If you got 3 days off after 3 shocks, you find some improvement. And again, with our shocks with interpolated rest, that's our most effective procedure. Fourth experiment here. What we're going to do is do 3 versus 5 shocks, all with interpolated rest. And under these conditions, we actually find some nice differences. So here, with the freezing data, there's our traumatic shock group versus the blue, which is a restraint control. And both the 3 and the 5 day shocks with interpolated rest actually perform equally well. In our shuttle escape latencies, what we find is that 5 days of interpolated shocks, that's the blue, the red triangles, actually outperforms the animals got three small shocks with interpolated rest. So more is better, but when the shock is when the preconditioning shock is relatively severe, then you need a day of rest in between. These are data that we published actually a while ago that I think are probably a really significant finding. This is from Miner and Sadi in 1997. Basically, what we're going to do is look at the benefits of post-stress glucose consumption on performance in this paradigm. So this is a 15-day procedure. On days 1 through 3, we pre-expose them to a glucose-sucrose cocktail, 40% glucose, 5% sucrose. Glucose is not really sweet. If you add sucrose to it, you dramatically increase the avidity for the solution. And you have to pre-expose them to the solution in this paradigm, or else on the day of stress, they actually will not drink it. On days 13 and 14, all rats were exposed to, oh no, actually this is wrong. This should be day 14, all rats were exposed to 100 variable duration inescapable tail shocks or restraint in tubes. And then immediately after that stress session, put them back in their cage. We put a water bottle on their cage, and it either has this concentrated glucose cocktail in it, or it has water in it. We do that in a 2x2 factorial design to create 4 groups. And then on day 15, we do standard shuttle escape testing. And here are the data from that experiment. These data are showing that during the pre-exposure days, everybody drank the same amount. This is the amount they drank. This is the amount they drank after the stress. So the restrained water is drinking water here. The shocked water is drinking water here. Shocked glucose cocktail is drinking glucose, the glucose cocktail. They always drink slightly more of the glucose, which is actually good for them. It's really good for them. But it's such a viscous solution that I don't think it's a lot of work to actually drink the thing. So we never get a significant difference between the glucose cocktail consumption and the other solutions. What's dramatic here is that this is our group that got the traumatic stress and water. What you see is for the group that got the traumatic stress and then the glucose cocktail, their performance in the shuttle escape task is equally good to all of the groups. So here, this is our measure of conservation withdrawal. It completely gets rid of this measure of conservation withdrawal. I'm not going to present the data here. We've since shown that it also gets rid of the exaggerated fear conditioning. So this is a very powerful manipulation that is really hormetic. So there were some procedures that we identified in the previous work, in the first work, that weren't effective. Okay, and what we're going to see now is whether we combine those procedures, those ineffective hormetic procedures, with post-stress glucose, whether that enhances training. And so one procedure that was really ineffective is when you gave them 3 days of shock, little shocks in a row before the traumatic shock, and they had water in between the sessions. We're now going to give them glucose and see whether we can improve performance. 3 days of shock with interpolated rest in water was an effective procedure, but not completely effective. And again, we're going to see if we can improve performance actually with giving them glucose. Here are the data. And what you see is everybody gets inescapable shock here. So the circles are the shock water group. Here is the diamonds here are the shock water. So the 3 days of shock with water following before the traumatic shock. And again, we see that nothing helps these animals. If you give them glucose, however, you find a dramatic improvement. If you give them glucose plus a day of interpolated rest, then you find dramatic improvement. And that same pattern is again reflected in the shuttle escape latencies. So here, glucose is again is improving, is taking ineffective hormetic procedures and making them hormetic. In this experiment, we push the limits. So what we're going to do in this experiment is actually give animals 2 days of trauma. So we do our baseline exposure to glucose. 10 days later, we expose them to, on each of 2 days, 100 variable duration inescapable tail shocks over two consecutive days. So all groups here get 2 days of trauma. And now what we're going to do is either hang a glucose bottle on their cage or a water bottle, so that we're going to create four groups. One group gets glucose on both days. So inescapable shock here, glucose, inescapable shock, glucose. One gets water following both. One gets glucose following the first session, water following the second. One gets water following the first session, glucose following the second. And what we find, so what we find here is for the freezing data, the group that got water following the 2 days actually shows a very exaggerated fear response. The group that got glucose following both days shows a complete improvement from that. Importantly, if you got glucose, the triangles here, if you got glucose following the first day, water following the second, you actually show dramatic improvement. That improvement is not clear if you got water following the first day and glucose following the second. And here the data are actually much clearer for the escape latencies. So here, the animals that got water on both days or water on the first day both show significant impairment here. If you had glucose following both days or glucose following the first day, then the deficit in escape performance is gone. And so to conclude, actually running time, so, repeated exposure to mild stress eliminates both the enhanced fear conditioning and the shuttle escape deficits otherwise produced by traumatic inescapable shock. The level of resilience produced by repeated exposure to mild stress may increase with the number of pre-exposure sessions. Interpolated rest days do not contribute to resilience when the pre-exposure stress is mild. Interpolated rest does contribute when the pre-exposure stress is moderate, with an alternation of stress and rest being the most effective procedure. The benefit of preconditioning with moderate stress increases with the number of preconditioning sessions. Post-stress glucose transforms ineffective preconditioning procedures into effective ones. Consuming glucose following traumatic stress protects against subsequent trauma. So traumatic stress under these circumstances actually becomes hormetic. And thank you for your time. I'll take questions.
Thank you. Ms. Frank, is now open for questions.
Yeah, I have a question. On the chart that's shortly before this, where you alternated the water and glucose, and one was water first and the other was glucose first.
Right.
And they did, one was had the rest of it, and one was way up with the water.
Right. So if you get trauma and you get water after that, all the damage from that trauma is already done. If I give you glucose, it's not going to help. Right? So in terms of treatment, if you have PTSD and I give you glucose, it's not going to help. But if you've been traumatized and you're at risk for developing PTSD, then if I give you glucose at that time, it actually should prevent the development.
So very interesting model. One is that you mentioned the blood sugar and the other one's glucose. There's a lot of idea that sugar's bad, and so it's not bad. What's it doing? Why is there a need for sugar?
So the question is, why do you need the sugar? And the answer is complicated. So here, what we can't do is do one of these nice things where we see an increased D-shock protein or something like that. We're dealing with a behaving organism. And so we need to look at the brain and how the brain functions during stress. And the brain actually changes the way it processes information, particularly under traumatic stress conditions. And in particular, the hippocampus stops functioning. And that's largely due to an increase in cortisol, which actually blocks the uptake of glucose into the hippocampus. And without glucose, the hippocampus doesn't function well. The consequence of that is you change the way you consolidate memories. And so the hippocampus provides a spatial temporal code, sort of a drawer in which you put memories. And without that code, it's very hard to find the memory. So if you try to treat a PTSD patient, you say, okay, sit down, let's talk about Afghanistan. They have no memory of it. Okay? And then they walk out the door and they have a flashback, and you're not there to help them through it, and things just get worse. So what we think that by increasing glucose, what we're doing is, first of all, we're bringing the hippocampus back online, and so memories get consolidated properly. We're also replenishing liver glycogen, which is depleted by this level of stress. And so a lot of the sequelae that occur in the brain as a result of trauma and too little glucose actually are impaired.
So the hyperglycemic?
Yes. So we know, actually we have to measure this in rats, we've done it in humans, which is actually much easier. So if you drink a glucose solution, your blood glucose levels will go up well over 100 within 5 minutes, and well over 140 milliliters per deciliter within 30 minutes. So they are hypoglycemic.
Are you suggesting that the memory consolidation may be mediated in part by sleep?
So the question is the memory consolidation mediated by sleep? We think so. We have a study planned in humans, but all we need is money.